My name is Greg Scherer. My lab at Stanford investigates the mechanisms that underlie pain perception, and how opioids such as morphine interfere with some of these mechanisms to generate analgesia, but also side effects such as tolerance or addiction. This study has been led by my postdoctoral fellow Francois, and has to do with endogenous opioid peptides known as enkephalins. And what Amory has discovered is that there is a neural circuit in which neurons in the brainstem can control the release of these enkephalins at the level of the spinal cord to modulate pain thresholds. And he's going to tell you more about these experiments now.
Hello, my name is Francois. I'm the first author of this paper. We couldn't have studied enkephalin without the proper tools, and co-author Adam Altman from Janelia developed a pre-pro enkephalin Cree mouse, or PNCree, so we can manipulate enkephalinergic neurons in the spinal cord. We first wanted to manipulate these neurons to see if enkephalin can contribute to pain modulation. We expressed the modified muscarinic receptor HM4DI in PNC neurons in dorsal horn of the spinal cord where nociceptive inputs enter the central nervous system.
Following inactivation of spinal PNC neurons, we observe a drastic increase in mechanical pain sensitivity, but surprisingly, no change in other sensory modalities including heat. To understand the mechanism by which this phenotype can occur, we use slice electrophysiology. While activating primary sensory neurons, we recorded from random interneurons in the spinal cord and used channelrhodopsin to activate PNC neurons and measure the effect on synaptic transmission between primary sensory neurons and spinal cord interneurons.
We observed that activation of enkephalinergic neurons can reduce synaptic transmission between primary sensory neurons and the spinal cord. The enkephalinergic mediated inhibition specifically targeted the delta opioid receptor in sensory neurons. This presynaptic inhibition of DOR-positive neurons will account for the reduction of mechanical pain that we observed with HM4DI. Interestingly, activation of PNC neurons release both enkephalin and GABA to produce presynaptic inhibition. GABA will have an immediate but short-acting effect, while enkephalin will have a delay onset but will act longer. At this time, we still didn't know how these neurons were recruited and when the presynaptic inhibition will occur. To identify the circuit responsible, we use retrograde rabies tracing from spinal PNC neurons. We observe that one area in the brainstem, the rostroventral medial medulla, or RVM, sends strong projections to spinal PNC neuron. This region has been implicated in descending control of pain, but curiously, the neurons that we identified in the RVM were all GABAergic and will form a desynaptic inhibitory pathway with the PNC spinal neurons. To confirm that GABAergic RVM neurons control spinal PNC neurons, and thus mechanical pain, we combined the newly designed retrograde virus AAV retro with HM4DI to selectively inhibit GABAergic neurons projecting to the spinal cord. In parallel, we expressed channelrhodopsin or halorhodopsin in GABAergic RVM neurons and implanted a fiber optic in the spinal cord to selectively manipulate the terminals in the dorsal horn. Activation of GABAergic neurons projecting to the spinal cord produces a similar phenotype as inhibiting spinal enkephalinergic neurons, an increase in only mechanical pain sensitivity, which confirms the link between these two populations of neurons. The pathway we identify raises interesting questions concerning how our brain can control pain thresholds. We revealed the pathway selective for mechanical pain, suggesting the existence of other pathways selective for other sensory modalities. We started to characterize how internal states may engage this pathway, but much more remains to be done to fully understand how emotions and other brain mechanisms interfere with our perception of pain.